All right, and thank you very much. Thank you for the organizers, and thank you for the exciting discussions we had so far. So what I'm going to talk about is about my group's work, about using computer modeling to design DNA, RNA, and protein DNA hybrid nanostructures. So we already heard great talks today from William, Faye, and Carlos about DNA nanotechnology and what it can do. And I'm on the modeling side, so I want to use computer simulations to help understand the function of these designs, and also use computers to design devices that are so complex you just can't come with empirical rules and do trial and error experimentally. Now, from a simulation perspective, these systems are extremely challenging. Typical DNA origami has around 10,000 bases in it, and concerns very rare events such as creating bonds and breaking bonds. So this is out of the reach for full atomistic molecular simulations, which typically only capture microsecond behavior, but we need to get to milliseconds, seconds, or even longer. To do that, the procedure is called coarse-graining. So you will give up some level of freedom. So you will replace all the atoms by a single superatom, which we call the coarse-grained particle. And we parameterize the effective interactions between these superparticles to represent the basic behavior which you need to accurately describe nanoscale system. In the case of DNA and RNA nanotechnology, we want to capture correctly the thermodynamics of the system, mechanical properties, and basic structure of DNA duplexes and single-stranded conformations. So over the years, me and my colleagues at Oxford University, hence the name OX, when I started as a grad student there, have been developing this OX-DNA model with these properties. Back in the day, we would be stretching single-stranded DNA, and we were excited how complicated system we can simulate, but thanks to the GPU computing and the progress it has made, now we can have 3 million nucleotides on a single GPU simulation these days. So our model is currently the only one on the market which can represent the strand displacement process, which William briefly introduced. It's the blue strand comes in, binds to the red strand, and kicks out the green. You can show that this simple reaction can realize arbitrary logic gate circuit. So if you can implement molecular logic at this level, you can do computations inside cells, for example, with this mechanism. And it's an example which requires a model which can really do base breaking and base formation very quickly. We have developed associated ecosystem around this simulation tool to make it easier for experimentalists to set up their system, run the simulations, and evaluate them. And also it evolves into a standalone design package which you can use to actually design these devices. So I will talk about a few of the components. So the center core of the analysis tools is called oxview.org. Talking to experimentalists, we realized that installing anything is always a barrier. So this just runs in your browser. You just go into oxview.org. It's written all in JavaScript, runs pretty smoothly. It can be either interfaced with existing design tools like Carlos's Magic DNA or CAD-Nano from Sean Douglas, where you can import existing structure into our tool, or you can also use our tools as a free-form designer. So in this particular example, we imported this molecular slide. So this nanostructure, this long stick, was designed using Magic DNA, I believe. And then we have positioned another origami around it in our tool by just basically generating 6 helix, multiple helices and connecting them together. So you can edit any structure you want. And you would ideally want to see how the structure actually looks like in the experiment. As it was mentioned, life is quite different at nanoscale level. We are constantly bumping to molecules and everything is jiggling around. So what you can do, you can interactively connect to our server based in Arizona. If you want to contribute to the global warming, we have a free GPU server there. You can copy over your design, it runs the simulation in the underground of the computer science building, pumps out the hot air into the already hot surroundings. And every 30 seconds sends back to your computer what is the status of the simulation. Now, typical structure to really characterize its entire conformational space takes maybe 1 to 4 days simulation, but within a few minutes, you can get pretty good idea, if the structure is crumbling or misbehaving from the design you originally intended. Now, for the people who want to do more complex stuff, we also have scripting incorporated in the system. So if you know JavaScript, which I always feel very guilty when I tell my students to learn JavaScript. It's like asking people to clean toilet with toothbrush. But it works very well. So this is a very simple code which says to every unpaired end on this 2D origami add single-stranded DNA of 6 bases. So this, for example, would be the way how to functionalize a breadboard structure to which you can attach proteins and other molecules, etc. It's hell difficult to do by just clicking on every 70 empty bases there, but if you just say, take everyone and add poly-T spacer to it in JavaScript, it will do it very quickly for you, and then you can export the structure for simulations. Now, for those people who need to actually characterize really entire conformational space of these structures, so we have also free server, also in Arizona, so also contributing to global warming. You can, for free, register there, upload your structure, it will run the simulation on GPU cluster for you, send you an email when it's finished, and it also has automated evaluation scripts. So the most typical design choices that we are asked by our experimental colleagues, we don't have bandwidth to respond to everyone, so some of the most typical things we just did over and over. You can just click and say, characterize the stability of the structure, characterize the flexibility, etc. It's all incorporated in this server. Now, it's all about also sharing, right? We have all seen the AlphaFold from Google success, which was only possible because of 150,000 proteins deposited in PDB database. So we would like to also create this cooperation and design sharing in the nanotechnology community. So we have established a server called nanobase.org, which is aimed at RNA, protein, and DNA nanostructure designs. Currently, we have 70 structures there. Whenever I'm giving a talk, I encourage people in the community, if you are reviewing a paper, please ask them to also upload after publication the structure to the database so other people can share it, edit it as they wish, or combine multiple of these nanostructures together to get new functions out of them. Eventually, maybe train machine learning algorithms on these structures to automatically generate functional structures for you from there. So now I would say those are the examples of the applications of our tools, but it's the other way around. We never develop the tools for the sake of tool development, but the tool is basically a side product of the 3 projects that I want to introduce. It's very difficult to get funding for just tool development alone, so all the stuff we have presented so far have been driven by experimental needs that we had when designing these structures. So example number 1 is done with Nick Stefanopoulos' lab at ASU. I didn't talk about it, but our tool also allows you to represent proteins. So you can just drag and drop PDB ID, it will load that protein, and you can design DNA nanostructure around it. I think by this time, everybody recognizes this is the spike protein trimer from coronavirus. And early in the pandemic, Baker Lab at Seattle designed this tiny yellow protein, which was designed to actually bind to the receptor binding part on the spike protein. It would inhibit its entry into the cell. The protein very quickly mutated, so while it was able in vitro to inhibit alpha variant to enter the cells, delta mutated so much that the affinity of this protein alone was so weak, it would no longer inhibit successfully. But if you position these proteins onto a DNA tile, which we helped to design, and specific design requirement was that these spike proteins on the virus exist in 2 conformations, which have different distances between the sites you want to block. So the structure needed to be flexible enough to cover both possible conformations, but you also want to maximize the affinity. So you don't want the structure to lose too much flexibility between bound and unbound state. So there has to be the right balance of rigid enough and flexible enough to cover these things. And in experimental data, which I'm not showing, but they should be on bioRxiv any day now, we show that this structure, due to the cooperative binding effect, was actually able in vitro experiment to inhibit the virus entry, while the single protein alone was not able to do it. Second application is designing photonic metamaterials. So these structures, if you were able to manufacture them, such that the cavity sizes would be comparable to visible light wavelength, so order of 100 to 600 nanometers, and build them in 3D, would basically act as a semiconductor for light. So this has been a huge topic in the community. We are very good at doing lithography in 2D, that's how CPUs are done, but we are not very good at 3D printing precise architectures. This is the reason why we are all here. But since we still don't have the 3D printer, the main goal how the community of the colloidal self-assembly people have been trying to assemble these in the past is by just self-assembly. designing individual blocks that just diffuse around until they bind. It has even been called the holy grail of self-assembly. It turns out that from all 3D lattices you can think of, these two actually interesting ones are the biggest bastards ever to try to assemble. Why is that? The reason is that any design you can come up with rationally always misassembles into something else. If you would position them by a 3D printer as you want, yes, you would have the crystal. But if you, for example, naively think, okay, this guy has 6 neighbors, so let's just say each sticky end binds to every other sticky end, and you start from the disassembled state and wait for the diffusion to do the job for you. Instead of getting the crystal, for this particular design, you start forming these long linear filaments. So in physics language, you would call it kinetically trapped state or alternative free energy minima, which is kinetically preferred. So you never get to the state where you want. So we develop an algorithm which very efficiently can search the design space of all the possible ways how you can design the interactions between the building blocks. And we call it SAT assembly. I don't have the time to go into the details, but happy to talk offline about it. The software tells us the minimum design needs to have at least 2 different particle types. This is their interaction matrix. Within a few hours, they assemble in the simulations correctly. So now I'm a theoretical physicist. I never held pipette in my life. And I developed this algorithm in the first year of my appointment as assistant professor. But when I approach different experimentalists, I would just hear jokes about spherical cows in vacuum. And they would not be willing to dedicate time to actually realize it experimentally, because I saw something in simulation. We even used the OxView, developed OxView primarily for this project to actually design DNA nanostructures, which was the best candidate material to scaffold these lattices, show them all these nice pictures, and they still send me to hell. So in the end, the only option was to get funding from DoD, for which I'm very grateful. Told my student who was a programmer that he's now an experimentalist, and it took us 3 years to build up the expertise, but we finally realized for the first time ever at nanoscale the particle lattice by self-assembly. And the third and last project I want to show is this, just got accepted to Nature Nanotech, should be published anytime soon. So one of the big directions I want to see the field go is combining the strengths of different approaches, protein nanotechnology with DNA nanotechnology, etc. So this is, to my knowledge, the first time we have successfully realized the DNA linear oscillating motor. This is done in collaboration with Fumble Club in Bonn. So we have this DNA hinge, which is connected by a double-stranded DNA. We cross-link a T7 polymerase motor to one of these arms. It binds to the start codon, starts transcribing the DNA until it reaches stop codon, and then falls off. So basically, it has periodic oscillating motion. So to connect this to the tasks of this meeting, when you are trying to do artificial ribosome, you need some energy processing, which will be pulling your single-stranded substrate on which you basically attach things. So this would be one possible candidate structure where, again, our simulations help to model the kinetics and operation of the system. And I can leave this movie play as it goes. So this is another, we have also generalized the model that we use to design the crystal to finite size assemblies. So this is from physics department Christmas party. So, I always feel very bad when I play this in the summer because it's not topical to the season. Basically, you draw a 3D object which you want to self-assemble. Then in the background, it runs the simulations and tries to find design which doesn't misassemble into something else. So in this case, the smallest design tells you need at least 6 particle types. That always assembles in the lattice. Then you can export this into actual 3D molecular simulation and run it to really make sure that it always assembles only the object you want and nothing else. So these simulations, despite them being very simple, still take 4 days to finish, but you end up with sort of Christmas tree looking forest. Right. So this is my group. The people highlighted in bold are the ones who actually did all the work. A big chunk of the work was done by Michael, who's a Foresight fellow. He was supposed to be here, but he's now looking for a kindergarten in Munich for his kids now, so couldn't make it. Thank you.
Thank you. He's missed dearly. Okay, question.
Question, a comment? Yes, over here. a lot of these structures that you talked about as being so difficult, I've seen them from Merkin and McFarlane, and so I'm missing.
Merkin or McFarlane never did cubic diamond nor pyrochlore. Those were different structures.
I have trouble believing that. They've published hundreds of different lattices.
And none of them was these two types because they are so difficult.
Okay.
What Merkin and McFarlane do, they do uniformly coated particles. But these structures you can show from the phase diagrams will never work from these particles. You need to have directional bonds.
Okay.
So they publish hundreds of structures, yes, but these two, which are the most interesting ones, that's why I call them bastards, because they can't be done with that approach.
Okay, thank you.
Maybe one more, if we have another one.
Fantastic talk, really enjoyed that. In terms of these multi-component systems, how much is the entropic term really weighting the fact that you can, in that you've got many more configurations, I guess it can form as it's getting towards the ground
So
state?
basically all these configurations we are forming have all the bonds occupied as well, but they are entropically preferred because for the 3D crystal you have to come up with the critical 3D nucleus, whereas the competing structure I showed is forming these filaments which are easy to form than the critical seed. Okay. And for the cubic diamond, which I don't have time to talk about, there the problem is different because the cubic diamond has the same free energy as a hexagonal diamond lattice. So any design which you form, which would form cubic diamond, would always switch between the two. So you form a mixture of the two lattices which doesn't have the photonic property. So the difficult part was, I didn't show the data, but we found a solution which avoids the hexagonal diamond and can only form the cubic diamond.
Again, multi-component, right?
Multi-component.
So it's not quite
You need
diamond.
at least two.
Yeah, interesting. So it's a diamond lattice with two.
Yes.
Two elements. Thanks.
Okay, well then I have our question of like, which architectures do you think this could be elevating? And I think you already gave a hint there.
Yeah, so I would say that for most of the structures that were actually mentioned in all the first two cases, the 3D printer and the breadboard, you always want to know that your design works, right? So you want to know that the proteins are positioned at the right distance. You want to check the thermal fluctuations of the system. You want to make sure it's not too flexible that it flips out and would like lead to some spurious interactions. So definitely for all these architectures, I would encourage people to first simulate them before sending poor students to do the experiments and optimize them in principle computationally. And this motor I showed, I think it could be a possible way how to bring some chemically powered motion into these systems. Like if you need to move along certain lines or bring in the building blocks in and the product out, this can basically provide directional motion nanoscale, which is chemically powered.
Wonderful. Thank you so much. This is
Thank
great.
you.